polymerase chain reaction. What it is? Polymerase chain reaction, PCR, is a method of making copies and increasing the amount of DNA or RNA. Through PCR, humans have the capability of retrieving the smallest strand of a genetic code and amplifying it over 1 billion times. How it works? The PCR technique is based off the process that a cell uses to replicate a DNA strand. An important component is the template DNA that contains the region of DNA sequence to be copied. Even something as small as a single DNA molecule can act as a template. The fragments needed for this to be replicated is the sequence of 2 short regions of nucleotides at either end of the DNA sequence of interest. Nucleotides are the basic structural units that make up the carriers of genetic information, which is DNA. The 2 short template sequences must be known, so the 2 primers, short sequences of nucleotides, that correspond to the template sequences can be synthesized. The primers bind to the template at their complementary sites and act as the starting point for copying. DNA synthesis at one primer is directed toward the second primer, resulting in the replication of the desired sequence. Additionally, nucleotides are necessary to build the new DNA strands. DNA polymerase does the building by sequentially adding on free nucleotides according to the instructions of the template. Uses of PCR. PCR can guide doctors to diagnose the proper treatments for illnesses and diseases. It allows medication to be distributed to the patients that actually need it. PCR has allowed for tremendous advancements in the scientific world, including research relating to the Human Genome Project, organ transplants, and paternity testing. PCR is used in the formation of genetically modified organisms, also known as GMOs. GMOs are food crops that have been altered using molecular biology techniques to enhance them with characteristics such as herbicide tolerance, pesticide resistance, greater nutritional content, and temperature tolerance. In the food industry, PCR is used for food labeling. Labels on foods containing dairy, protein, or any other potential allergens can help keep consumers safe. PCR has some defects, including the production of inaccurate results due to contamination, the difficult interpretation of the results of the reaction, and the explosive increase of genetic copies. Despite these disadvantages, there is always room for improvement of this technique that has been a huge advancement in the world of molecular biology.
Created using Powtoon.